Hi, in this video we'll be introduced to some of the most basic facts about biology. As you can see, I've put universal in quotes because there's almost nothing in biology that is truly universal. But there are a great deal of things that are true for 99.99% of life, and those are the things we'll try and talk about here. We'll discuss that DNA is the primary repository of information for life. We'll talk about the way that information comes to life, a fact considered so fundamental that is referred to as the central dogma of biology, which is DNA makes RNA makes protein. We'll see that DNA is organized into genes, that genes can code for proteins, which are made from the DNA via the intermediate molecules mRNA, and that mRNA is interpreted by tRNAs and ribosomes. And finally, that we share parts or genes with the simplest of life forms, which indicates that our genes are evolutionarily related. So in this course, we'll spend a lot of time talking about the three major types of molecules and cells. DNA, which was, as we have said, is the repository of information. Proteins, which are the structural elements and the molecular machines of the cell, the parts of the cell that do things. And you could say that RNA enables the transition between the two, a chief operating officer of the cell, if you will, as it decides which DNA information should be selectively converted into protein in which cell, although more and more functions for RNA are being found all the time. So all the information is stored in DNA. It's stored in a linear code of bases C, A, T, and G. And this code contains all the information to convert a single cell into a fully adult human being with approximately 10 to the 12th cells. DNA is double-stranded, and it's in the conformation of a double helix, with the information-containing part, the bases, in the center of the double helix, and on the outside there's a sugar phosphate backbone. Information contained in DNA is passed on by cells by the synthesis of new strands of DNA. The original strand of DNA is used as a template for guiding the incorporation of new nucleotides into a growing strand of DNA. As you probably already know, C's and G's always base pair with each other, and A's and T's always base pair with each other. DNA replication is semi-conservative, which is to say that each of the new daughter strands gets one of the original parent strands, and each gets a newly synthesized strand, depicted in a tan color here. Now, the central axiom of biology, which was historically named the central dogma of biology, and that is that DNA makes RNA makes proteins. And this is the direction of information flow. DNA replicates itself in a process called replication. Portions of DNA are converted to RNA in a process called transcription, and the mRNA is converted to protein by a process called translation. So the information stored in the sequence of 4 nucleotides is converted to a string of 20 different amino acids that make up all proteins. When we are going to turn a particular gene on, part of the DNA information is going to be transcribed into RNA, which makes multiple copies of a portion of that DNA gene. Here we see that the sequence of RNA is a match to the DNA sequence. Blue, red, yellow, green, green, copied into RNA. Blue, red, yellow, green, green. As we'll see in future videos, the RNA molecules are copies of the coding strand of DNA, and they are synthesized using the template strand of DNA. Proteins are the molecular machines of the cell, and they also have structural functions. But in this example, we see an enzyme called lysozyme cleaving a polysaccharide into smaller units. The portions of the protein that perform the biochemistry are called catalytic active sites. Proteins are folded in a precise way so that the constellation of reactive amino acids are brought into close proximity in order to effect this biochemical transformation at rates that far exceed what would be possible at room temperature without enzymes. This slide shows tRNA molecules. These are the adapter molecules which convert the nucleic acid code into the identity of an amino acid, which will then be incorporated into a protein, as the instructions are read on a strand of messenger RNA. So this mRNA molecule depicted here actually extends in both directions. The tRNA base pairs to the RNA, just like two strands of DNA in a double helix, assuring the proper positioning of each amino acid. On this slide, we see the steps of protein synthesis, and in particular, we have a 3 amino acid polypeptide chain that's going to grow into a 4 amino acid polypeptide chain. The ribosome will cleave the bond between the amino acid and the tRNA molecule in the P site, attaching it to an amino acid at the A site of the ribosome. You'll learn these site names in future lectures. After switching the connectivity of the polypeptide chain, the ribosome, the large subunit, will translate one codon. A small subunit will also translate one codon to get back into register with a large subunit, and a new amino acid will enter into the A site, and the process will be repeated. Although it's a simplification, there is a sort of correspondence that's often talked about between one gene and one protein. On your DNA, you have genes arranged in a linear pattern. Here the genes are depicted in red, with small yellow non-coding segments separating the genes. We'll see later on that genes can actually produce several variations of a protein, but that's a finer point for later. Organisms can have a wide number of genes. The smallest genomes of free-living cells contain about 500 genes. Humans have somewhat greater than 20,000, and in fact, we'll usually use the range between 20,000 and 25,000 genes. We don't know the exact number yet, but it's probably somewhere in that range. The number of genes themselves is not necessarily an indicator of organismal complexity, though it's somewhat of a rough guide. This slide highlights that we share many molecular similarities with simple single-celled baker's yeast, which indicates that we are evolutionarily related, and features that are important tend to be conserved over very large evolutionary timescales. When discussing evolution, the DNA record is the molecular fossil that you should want to look at. Whenever someone is speaking to you about the fossil record of bones as either proving or disproving evolutionary theories, 99% of the time you can just stop listening because they're not talking about the most relevant information. Talking about bones and fossils in the 1800s was certainly acceptable. Back then, we weren't aware of the information that bore on this relationship, but here you can clearly see the relatedness in the DNA sequence between humans and baker's yeast, and the relationship between these proteins is hard to ignore. The evolutionary relationships can also be exploited to give us information about protein function and the relatedness of different organisms. On this slide, we see a phylogenetic tree comparing a number of proteins, myoglobins and hemoglobins, and the point being made is that the sequences are more related for all of the hemoglobins than they are to the myoglobin species proteins, even though they have somewhat similar functions in that they bind oxygen. So the more closely related the sequence is, the more likely it is that the protein's function is related. So this concludes a whirlwind tour through some of the basics of biology, and I look forward to filling in the details for the rest of the semester.